Okay, we're going to be looking at the sliding filament theory. So the sliding filament theory explains how our muscles contract to create movement and pull onto the joints. Yeah. So if we look over here, this is one sarcomere. So a striated muscle cell is composed of the skeletal cell and then bundles of muscle cells in between. And so each individual one is sarcomere. So we're going to look at exactly what a sarcomere is composed of, and then also how the protein molecules work together to cause movement. So a sarcomere is basically like two strings with some elements holding them together. So we have two filaments. We have the thick filament. We have the thick filament and then the thin filament. The thin filament is a protein molecule called actin, this one, and the thick filament is the one called myosin. And together, because the sarcomere is composed of so many of these fibers together, they form Z discs in between. So the sliding filament theory basically explains how during movement, these Z discs move closer together for the muscle to contract and release. So now we're going to go through the process of how the sliding filament theory actually occurs. So when your body wants to move, there's a nerve impulse that's sent from your brain, and it sends an action potential through your spinal cord into wherever region of the body the movement will be happening in. So the action potential travels through the neuromuscular synapse, and when it reaches the junction, it releases acetylcholine. And acetylcholine is a neurotransmitter, and the release of acetylcholine stimulates the T-tubules and the sarcoplasmic reticulum to release calcium. So the first step is really just the kind of activation of the structures, right? So there's synaptic transmission, and then the action potential travels through, and then the acetylcholine is able to stimulate the sarcoplasmic reticulum to produce calcium. So then the calcium molecules are now just in the muscle, right? And the calcium molecules kind of float over, they're just, in the air. And here we have a thin filament. So remember that a thin filament is really just actin by itself, right? So if we go back and look at the structure, the thin filament is only this section. So right now we're not looking at this section. We're going to ignore that and only focus on the thin section. So the thin filament is composed of actin and then also tropomyosin and troponin. And these are just two protein molecules that work together to ensure that the structures move as they are intended to. So the calcium that was produced earlier travels over and it binds to troponin. So this is molecule number 1. Right now, troponin is kind of holding on to tropomyosin. The two of them are attached, they work together. Calcium binds to troponin first. Then the binding sites are open because as calcium binds to troponin, troponin then opens outwards, pulling the tropomyosin off of the actin filaments. So if we look before here, the tropomyosin is kind of like a rope that covers, or like a door that covers the actin, right? It's on top of it and it blocks the actin. When the calcium binds to troponin, it's like pressing a button and making the tropomyosin move off of the actin. So now the actin is empty, there's nothing covering it, and the binding sites are exposed. This is where the thick filament, also known as myosin, comes in. So both myosin and actin have binding sites on their exterior end to ensure that they are able to bind and hold together. If we look at the structure of the thick filament, so of myosin itself, it kind of is composed of a lot of fibers with club-shaped heads at the end of them, because the binding sites occur here. This part is elastic and it allows the movement of the stretch, and when this part binds to actin, it is able to move and do everything else. So now that the myosin and the actin have binded, what basically happens is they form a crossbridge. And one important thing to note here is that there was already a hydrolyzed ATP within the myosin head. So right here there was already an ATP separated to ADP and an inorganic phosphate. So as the two of them bind together, the myosin filament is going to release this additional phosphate group, and that produces energy. And this energy causes this crossbridge. We call it a crossbridge because if this is the myosin filament and this is the head, the actin part that it binds to is not perpendicular. It's like at an angle, right? So this is a crossbridge. And with the release of that inorganic phosphate, that provides energy for the myosin head to then pull the actin filament over. So once again, this is binding, releases the phosphate, and this is pulled this way, right? And then we have to remember, this is just kind of a simplified diagram of what's actually happening because there are lots of these filaments like attaching at the same time. So the ending result is if this myosin, because these are all the same things, there's just more of them now, binds to this actin and it pulls this over, then the Z disc is moved in, right? And that's what causes muscle contraction because you're opening and closing and opening and closing. Now at this point, after the contraction happens, there's no longer, there's no longer a crossbridge because the myosin is attached to the actin, but now they are perpendicular because the filament has moved over. So how the filament attaches is a new ATP molecule binds to the head of the myosin, and that's what causes the detachment, right? And once that ATP molecule attaches, the myosin head detaches from the actin, and the ATP is then hydrolyzed into ADP and phosphate, and it's ready for another crossbridge formation. So the next time this occurs again, the myosin head will be able to release that hydrolyzed phosphate group and perform a power stroke to pull the actin filament over. And that is really everything from the sliding filament theory. So the most important points to remember is just that without the attachment of ATP, myosin head will always be connected to the actin filament. And then the power stroke, which is when it pulls the actin filament over, is activated when that inorganic phosphate group is released. And that's what causes your Z discs to move towards each other and cause muscle contraction to happen. Yeah, that is everything for sliding filament theory.